CRISPR is a gift from Mother Nature to all of us. It's like sunlight or oxygen or trees. It just occurs in the world. Jennifer Doudna's amazing discovery was how to take that gift and make it something useful for all of us.
Dr. Jennifer Doudna is an internationally acclaimed biochemist based at the University of California, Berkeley in the United States. Dr. Doudna broke onto the scene in 2012 with the discovery of CRISPR-Cas9, a genome editing tool which continues to transform our future in a profound way.
The story of CRISPR-Cas9 is a story of curiosity-driven science that went in an unexpected direction. In my case, I was working with a collaborator, Emmanuel Charpentier, to understand a bacterial immune system called CRISPR. And we figured out that a protein in that system called Cas9, we call it CRISPR-Cas9, is a protein that can be programmed to recognize and cut DNA in cells. And by cutting DNA at precise positions, we can trigger changes to the DNA sequence.
Genome editing is not a new concept in science. However, prior to CRISPR, it needed the highly specialized skills of a scientist to alter the DNA sequence of a living organism, and 1 to 2 years of waiting for the results. With CRISPR, however, the process has been made easier and faster, taking only 3 to 4 weeks to see the results. This cost-effective and more efficient technique has opened up many possibilities that were previously thought to be impossible.
Well, you know, the population worldwide is not only getting bigger, but it's getting older. And I think that the types of diseases that we will confront as humanity will change. We used to think about only treating a disease, not curing a disease. I think that CRISPR has the possibility that you could actually cure a disease.
The technology of genome editing is important, not only for understanding human diseases, but also for curing them. The DNA that causes genetic diseases such as sickle cell anemia, muscular dystrophy, cystic fibrosis, and possibly cancer can in principle be corrected or altered by using genome editing.
Perhaps the scariest ones, like cancer and neurodegeneration and perhaps diabetes, will become really, really serious in the next 10, 20 years. And so I think that this type of technology like CRISPR-Cas and other related technologies are going to be revolutionary in helping us to live, not only live longer, but live better. And I think that's critical.
This will be the century, I am convinced, when Jennifer Doudna's discovery of what CRISPR can do will be thought of as a century when we've gotten rid of genetic disease. I know that sounds optimistic, but think back to the 20th century. Humanity eradicated polio. Humanity eradicated smallpox. This century will be the one when we, if not eradicate, we take a serious stab at eliminating genetic disease the world over.
Apart from biotherapeutics, another way that CRISPR can contribute to the betterment of mankind is through agriculture, which causally is essential to supporting a growing world population brought on by improved healthcare.
The planet is growing. It's getting warmer. What do? Well, the answer is we create animals and crops that serve the needs of a growing and warming planet. That is already happening, and in 50 years, I really think that agriculture the world over will be done using CRISPR.
I think in the future, there will be a large global impact of genome editing to create plants that are resistant to pests, to things like climate change, and also to make plant products as nutritious for human health as possible.
Despite its potency, debates have heated up over the ethics of how this technology should be used. Genome editing on human embryos and genetic bioenhancement were, among other topics, the most debated ones.
I don't think there's much debate about the usefulness of CRISPR-Cas9 technology in the human clinics, as long as you're dealing with adults. I think the real ethical, moral issue comes when you're trying to deal with human eggs and sperm, which is what we call the germline, because now you're dealing with multiple generations and you can't predict what's going to happen.
I've often asked myself, what is the best approach to bioethics? Is it to allow specialists who work in the field to study the sort of the outcomes of science and try to prescribe directions for science, or is it for scientists and others who are engaged in the work directly to be involved in that conversation? It's been very important to me to be involved actively in that conversation internationally, to be encouraging discussion about the use of genome editing, and to be thinking with my colleagues, my collaborators, and my fellow scientists about how we ensure safe deployment of genome editing in humans, plants, and animals.
There is no technology that is universally positive. Nuclear energy can be used to create power plants to provide light. But nuclear energy, we all know, has other uses as well. My biggest concern about CRISPR is that it will be used for purposes to improve human beings in a way that would be unethical and dangerous. Should I be using CRISPR to have a baby with desired facial features? Or should I use CRISPR to make myself better in some way? I cannot tell people what to do. I am concerned about this.
Curing genetic diseases and adapting our food production in the face of global warming, these possibilities are already on their way to becoming reality. But this does not just rely on scientific discovery alone. It also requires the sharing of knowledge with other scientists and with the younger generations who will be the stewards of our future on this planet.
It's important to me to be contributing my efforts and my energies to future generations of people, and frankly, to also ensure that everyone has a chance at a great education. I think that it's very important that young people see that our society values science, values scientists, values diversity in science, and that frankly that scientists are not, we're not all geeks with glasses and hunchbacks, but that we can be vibrant people who have lots of other interests outside of the lab, but we have a real passion for understanding our world. Science is a process. It involves asking questions and figuring things out that nobody has investigated in the past. And that's what I love about it. It's a process. It's something that can't really be predicted. So I think that for me, what I get excited about is to try to figure out what the most important questions are that I and my colleagues can be going after at this point in time, and of course, importantly, how we can use our results and our data to help advance science and technology for the betterment of human society.